Very good morning, students. I hope you're all very excited to learn more about proteomics. And as you've heard in some of the really distinguished speakers who have talked about the growing field of proteomics, that is really one very exciting time for us to be part of this journey, where we can see how the new and new tools are being developed, but also more importantly, how we can utilize these tools to understand the biological questions or a lot of clinical challenging questions which have not been addressed yet. And that's where you can see the power of these technologies, right? And also I think, as a grad students who are all interested to do research, I think it's important for you to start thinking about that, how you can present yourself well, how you can illustrate your ideas well, how you can show your data well. And that's where data representation, different type of pathways analysis, network analysis, making those drawings becomes very important. I think you have heard one of the speakers, Beth, she talked about how you can visualize and make the representation of your data. It's not only about doing good science and good research, but also how you can make it effective to present your ideas to others who can also understand in more simplified way. And that's why we are introducing you to many tools and softwares and different type of programs which you can utilize to really illustrate your data, to bring to the connect with biology, as well as also to illustrate the significance of your findings in a very lucid manner. So let me share my screen and start today's lecture now. So we are discussing about quantitative proteomics, and I'll be continuing on that today, along with some network analysis, which we'll be talking. So first, I'll give you some quick recap, mainly focus on quantitative proteomics and applications. We will continue where we left on the pathway analysis of Reactome, and also connect with the Gse analysis, which is gene set enrichment analysis with the Reactome, and then also show you how to use the string for the network analysis. And then the quiz will be based on the Reactome, for which you already have files and data with you. So just to give you a recap, we have been discussing about different in vitro labeling methods. Especially we discussed about isotope coded affinity tag and isobaric tag for relative and absolute quantification, and TMT tags. Some of those were the label based methods. We also talked to you about label free methods, and we have been discussing about how mass spectrometry, which has become very important tools in field of proteomics, can be utilized from the protein identification using MassQuant, or protein quantification analysis using MaxQuant, followed by visualizing the data in the secondary data analysis from MetaBanalysis, Perseus, and now looking at more of the pathways and network analysis. So this is where we are leading right now. We are still focusing on only one of the key technology, which is mass spectrometry based proteomics. So let's just change the gear and now discuss about some of the in vivo labeling methods of quantitative proteomics, especially people who work on the cell lines, cell cultures. Let's say you're working on the bacteria, you're working on the yeast, you're working on any other cell models. Then you would prefer to use in vivo labeling methods. And there are different type of methods people have tried, especially labeling with 14 and 15 and media, or culture derived isotope tags. But one which has become very prominent and very popular is the stable isotope labeling by amino acid in cell culture, also known as SILAC. We'll focus mainly on SILAC because SILAC is widely used for the doing the cell culture based experiments. Now, as some of you are exposed in this area, some of you may not have done any kind of cell culture work, but the idea is when you are growing the cell, you are providing the nutrients to for the cell to grow, right? And you have to keep those cell in the right temperature condition, right nutrient medium conditions, which incorporate sugars, amino acids, everything which can nourish the cell. And then you have to grow the cell for a few days. After that, you have to change the media. Again, this passage, you will now go for the next passage, so that cell again continue growing. And this way you are going to maintain your cell for going to multiple passages. While doing so, like, if you are interested to look at any specific biological question, for example, you want to look for the effect of a given drug on the cell, after certain passages, you can do the drug treatment on the cell. One is non-drug treated, one is drug treated. And now you can see what is the effect of the drug in the cell after a few hours time, or after a few days time, after a few weeks time, or you're looking at the effect of a given specific stress condition on the cell, you can do the same type of experiment. So people do plan a lot of cell-based experiments, and that's where the SciLec becomes very important, because it relies on the concept that the media which you are using to grow the cell, that media itself can have a minor change. So in the media, so let's say you have two conditions to compare. One is no drug treatment, one is drug treatment. So now in both of those, you are going to grow the cells in the medium, and that medium will contain amino acid mixtures. One of the amino acid is going to be light, and one of the amino acid in the other condition can be heavy labeled. So in this way, now you are introducing the variation which can be utilized by the cell to incorporate that, and then eventually when you extract the protein from it, then you can use that information in your mass spectrometry to look for the protein quantification. Now, since we are using the labeled analog of amino acids, so all the amino acids are going to be same except that one amino acid which you are changing, either light form or the heavy form of that, which has to be incorporated in the cell, and you are allowing sufficient time for the cell to grow, and you are giving 3, 4, 5 different passages, so that after sufficient days and time, the cell totally incorporates this medium into the cell, right? So now it becomes part of the cell's metabolism, and now you will have all the amino acid which will incorporate either the light or the heavy forms which you have after the number of cell divisions which you will allow. So let's say you can take arginine as one of the amino acid, or lysine, which is having 13 carbon atoms, and you will introduce the light form or the heavy form of that. Just to give you an idea about the cell culture based experiment and how you can use SciLec here. So first you need to prepare the SciLec labeling medium, which usually comes commercial now. I think there are different labs, different companies, Cambridge Isotope and others, which provide this kind of formulations of SciLec labeling medium. First, you are growing your cells, let's say in DMEM medium, which is the standard medium of Delbeco modified Eagle medium, where you will provide the nutrients to the cell, and then you will change those cells to move to the SciLec labeling medium. And now let's say you are going to have two conditions, one for the no drug, one for the drug treatment. Then you are going to apply those differential treatments to these SciLec cells. After 4 or 5 passages, when the cells have already incorporated this SciLec medium, then you are going to do the cell lysis, protein estimation, and then your rest of the analysis of mass spec and quantification becomes straightforward. So major innovation lies here is in the introducing the variation in the beginning condition of the two different conditions. So let's say 13,6 L-lysine, which is the stable isotope of 12 carbon 6 L-lysine, and that shows 6 Dalton change. So if you can use this 13C6 or 12C6 form, that can be one of the pair which you can use, or you can use even arginine, another commonly used amino acid label, where again you can have 13C6 L-arginine or 12C6 L-arginine, and that is also going to show you in the mass spec data for the same peptide. You will see the 6 Dalton change in the because of the heavy and the light forms. So for doing this kind of experiment, of course, I can share you a protocol paper which will be more detailed in manner if you are really keen to work into the SciLec and this type of experiment, where you know to optimize many conditions, and there is a good nature protocol paper which is available for doing that. You are looking at your light, medium, and heavy light form and heavy form, and you are giving sufficient time for cells to grow and incorporate this medium, this amino acid in the cell. And for doing that, after each passage, you have to do mass spec analysis so that you can see whether the labels are incorporated completely or not. Once you see they're completely incorporated, then only you are ready for your actual experiment. So now, just to give you a nutshell, that we talked about two different conditions to be compared, where you can have the light forms of the 12C6 arginine or heavy form of 13C6 arginine into the medium. Each of these cells are going to grow for 4 to 5 passages. After that, they should have incorporated the amino acid from the medium, and then now you're going to combine both of them, lyse the cell, and after that you are going to run them in the mass spec and see can you monitor these 6 Dalton change for each one of these peptides, and you are going to use that for the quantification information. So it's very powerful technique because you are not going to do anything chemical treatment like what you do in the eye track or what we have seen in TMT labels, right? You are actually adding a separate tag on the peptides, which is not the case here. You are actually giving the natural amino acids, which you are allowing sufficient time for cells to incorporate and take those labels. So you're not going to expect to see any chemical difference between these tags, which is being seen earlier. So ideally, you are going to have the cells now, which is like normal cell, and you are giving sufficient time for cells to grow and intake this SILAC light and heavy form of the tags. So they are going to really behave like a control cell population. Everything is same except that one amino acid which is changing. So that is not the major change in the cell overall. It's quite efficient, quite reproducible. In fact, the labeling efficiency could be as good as 100% because you are, even after 4 passages, if you see it has not completely incorporated, you can again change the media, again add the SILAC media, and now you can grow one more round, one more passage, and see now whether it has incorporated 100%. So it is very efficient and quite reproducible, and stable isotope labeling could be almost 100% in this case. You are going to do the rest of the step like earlier TMT or iTrack, where you will mix the samples so that you have now the two conditions which you are comparing. They are already having different type of tags, and they are going to be mixed together so that you have the less steps, and handling errors could be minimized so that your control and treatments are given same set of treatments and you are not going to change the things. There are new set of SILAC labels now, which can actually go in 5 multiplexing conditions to be compared. I talked to you earlier for iTrack, which can do 4-plex or 8-plex. We talked about TMT, which can do 6-plex, 10-plex, or 14-plex. Even SILAC could go even more of the 5-plex. I have not followed very recently, but it could have gone even further to that. So idea is that you want to really look for various type of cell culture based work and look at the quantification. Disadvantage could be that it's mainly applicable for cultured cells. You can't use for the solid tissue and the body fluids, although there are some new variations of SILAC, super SILAC is coming, which is trying to overcome these limitations. But major bottleneck is if you are not into the culture based work, it's quite time consuming and quite skilled work. So everybody cannot do that. So everybody cannot make use of SILAC because unless you are really good in doing cell culture work and making multiple passages without any contamination of the cell, you can't use this type of quantitative proteomics strategy. But there are a large number of labs who do work in the growing bacteria, growing yeast, growing different type of cell cultures, cell lines for cancer and other diseases. All those can be heavily benefited by using SILAC based methods. Again, the choice of heavy forms of the amino acids are quite limited. Therefore, the number of states which you can compare can be very limited as well. Also, when you are doing the labeling of arginine amino acid, that also sometimes shows metabolic conversion to the proline in the cell culture media. And that actually poses some challenge because proline, as you know, is one of the more unique amino acid with the more cyclic form, which creates different problem in the tryptic digestion. So therefore, again, arginine proline conversion also has to be checked while doing the your SILAC based experiments, and that is some software tools now available which can actually look for this aspect of how much conversion has happened, and you can try to minimize those interference in the labeling process. So some of these could be limitation, but the applications are enormous. As I mentioned, if you're working on any of the cell lines, HeLa cell, different other cancer cell lines, this can be very well utilized. Your media formulation, growth optimization, everything, whatever you are doing for the cell culture will be same, except that you are going to change the SILAC medium after some time, and that will introduce the 2 different variations in your differential proteome experiment. This application has really worked into the cell signaling studies, looking at the events study for the protein complexes, various time frame analysis of temporal dynamics, because that's very important. If you're doing cell culture based experiment, you want to look for the cell, how they are behaving differently after drug treatment in a short to long term. So you are collecting, let's say, samples of a different time interval, and that can be very good experiment of looking at temporal dynamics of the and even the kinetics of those incorporation of these labels and what changes happens at the overall proteome level. People have shown the power of this technology for identifying the kinases and its substrates, looking at even differential membrane proteomics, right? So a lot of things can be done, as you can understand and appreciate now. This also adds a new tool in your kitty to look for quantitative proteomics. But finally, it all comes to how we can utilize many of these tools which we are talking to the actual problems. And as I said, idea here is to give you the confidence and the sense that the two things are not isolated. The technologies and tools and softwares and biology, these two should not be kept isolated. You cannot just keep working on knowing the workflows and developing technologies and softwares. You have to think about how well you can integrate that with the biology and clinical questions. And that's where I think I'm trying to keep giving you the idea. Not only we generate data from instruments and do the software analysis and the output of that, but also giving you some visual sense that how one could utilize that for the research activities as well. So let me take you to a quick overview of one of the projects which we are doing in the lab on the brain tumors, and I'll give you some idea for how quantitative proteomics have helped us to at least address some of these questions. Of course, given the time, I may not be able to cover a lot of these details, but at least want to connect you that what we talk on the different softwares and tools and overall workflows are towards these type of problems, why we need to utilize so much time for learning the technologies and addressing the technology needs if we can't look into the biological questions. So one project which we are working on, especially a much bigger program to say, with Tata Memorial Hospital, where we are looking at different type of brain tumors of glioblastomas, meningiomas, medulloblastomas, different type of brain tumors. And these are, as you probably are aware, these are one of the most deadly tumors given in the brain origin. And it's still a lot of unknowns are there, a lot of challenges are there for the identification of early disease to looking at the better therapeutics. All of these are the major challenges. And so one of the tumor which we work on is the glioma, which is having 4 different grades, grade 1, 2, 3, and 4. And from the histopathology, they can only make some sort of sense that, okay, how the disease looks in the different cells in the different grades. But still, there is not very clear understanding about what are the molecular compositions of these, how one could try to understand and control this problem. And much more so in the glioblastoma, which is another type of tumor which is quite heterogeneous in nature. Even in the same tumor population, if you see here, doctors use this particular reagent known as the reagent 5-aminolevulinic acid, which can show the fluorescence in the red light, and that shows a lot of heterogeneity within the same tumor type as well. And aim is again to understand what is that contributing factors for these heterogeneity. Also, we are looking at meningioma, another most abundant brain tumor. So just giving you feel that these are the most challenging type of tumors to work with. But when it comes to the experiments, now you can start thinking about your workflow, which we have been discussing. So we get tissue samples of these brain tumor patients, we get the serum sample, and in some cases, occasionally, we also get cerebrospinal fluid samples. So when you get these kind of different grades of the patient, different type of tumor samples, then we try to first do deep proteome profile with the fusion and QE, these kind of orbitrap mass spectrometers, using label-free quantification or label-based quantification or data independent acquisition-based methods. Also, sometime with the body fluids like serum and CSF, we use protein microarrays, which will be coming soon to you, and we'll talk about how this works, the technology works. Finally, you get a lot of leads and targets. You want to validate how many of those could be realistically taken forward, and those you can do using triple quad mass spec based SRM or MRM or PRM based methods, or one could also use ELISA or other type of immunoassays. So this is kind of general workflow. Now problem is bigger, but the reality on the technical side is this is the kind of workflow which you can follow for various type of the sample type. So just to connect you on the disease part and the clinical part of it, like why we are putting so much time and investment on looking at these technologies are essentially, let's say in this case of meningioma, when clinicians have some idea from the WHO classification that the tumors could be low grade to high grade 1, 2, 3, or when they look at also the radiology images, they find the location of tumor could be different. Some are in the closer proximity here in the skull base, some are in the supratentorial location. So again, the tumor locations could be different as well. So there are 2 different ways of classification. One is looking at the immunohistochemistry based and WHO classification for the grading. Other could be radiological images, which tells us the location of the tumor. So now we get all this information for every patient type. And then idea is when we get a sample, that sample has both the information of this type as well and this type as well. But for us, we are going to process each sample independently and generating with the label-free quantification methods its own peptide library. So for us now, we get so much data for every single patient with the at least 2000 plus proteins, unique proteins. And then we can analyze their data in different ways. Now, in this case, we are looking at how the radiology classification has classified these patients. Like, some patients are skull based, some are supratentorial. These are more aggressive type as compared to these types. Now this much information we have. So now we have data for each patient, and now we can analyze that data using different type of classifiers. So this time we are looking at only one variable, skull based or supratentorial. And the same workflow which you have seen now from the LFQ data analysis using MaxQuant, followed by MetaboAnalyst, we are able to come up with the signature of the protein which looks quite different. There's still quite a few proteins to narrow down. Then some machine learning approaches being used to find out what type of networks are driving this, and within these networks what could be most influential factors which will be very crucial for driving this aggressiveness of these type of skull based tumor type. And it came out to be like, fibrillin 5 was one of the very interesting target, and some of the EGF fibrillin like ECM protein receptors, those were very interesting targets. So those are kind of things which proteomics can move you towards this direction. So from the very unknown knowing about these patients, now we have started looking at dissecting out their molecular signatures. You're looking at the differential patterns or the signature patterns in these patients, and then you are trying to see which could be the most important driving factors which might be contributing towards these aggressiveness type. Also, once you get this data, you then start looking at the pathways, like which kind of pathways are driving these tumors. And looking at those, we start getting some idea that, there is quite a bit change happening in the two different type of skull base and supratentorial type, and, extracellular matrix organization or different type of other membrane trafficking type of pathways are driving this type of tumor heterogeneity and the differential behavior of these tumor type. Now from the same data set what we had, we also have idea for the patient's grading, like the patients are grade 1, grade 2, grade 3, those kind of tumor type, right? So now we are reanalyzing the data. So just think about, we have already acquired the data from every patient. We analyze the data, same data, in one manner using just the radiological classifiers. Now second thing is we are analyzing that data with the WHO grading and immunohistochemistry based data. So you are seeing the power of the LFQ based workflow or DIA based workflow, that you are not repeating your experiments, but the same data could be reanalyzed based on different information which you have. So this is now we are analyzing the data in the second different manner, using the grade wise comparisons in mind. And now grade 2 looked in this case quite homogeneous, but grade 1 shows quite differential response, as you can see here. And we also use different type of control to compare arachnoid and dura mater. These are from different regions of the brain, supposed to be control regions, and they're also showing differences because these are from different origin of the brain. But the grade 2s are looking quite homogeneous, except that one patient. But the grade 1s are showing quite a bit of distinct subtypes, and that's what we see in the heat map here as well. It means grade 1s are all not same. Like within the early grades of the tumor, some patients are having different clinical features which makes them different than others. So this kind of observation we can get from the proteomics, which was not possible to see earlier. And looking at the entire major landscape of the what is changing in this patient, and that's where you are now being exposed with the Reactome and the string and Gse analysis. You will see yourself that from the same data now you can start getting these kind of dynamic pathway analysis, and that actually led us to find out some very crucial factor like ILK integrin link kinases, PI3 kinases. Some of these are the pathways which are very much crucial for governing the meningioma disease pathobiology. Then we thought about taking this kind of study forward towards more patient-derived cells and doing some inhibitor study to see can we inhibit these pathways and see what could be effect of those. Again, I'm not talking a lot about biology right now. I want to make it use this information to just give you illustration how your data and pathways and your different network analysis can lead you toward the right path of making a hypothesis and driving your clinical and biological questions. So with the inhibitor, then we are able to see good inhibition based on the CPD22, one of the inhibitor which we used. And this kind of study is ongoing to see how best we can use different type of drugs and inhibitors now to find out what could potentially control these type of aggressive tumor types. So that just gives you the idea that same data set of proteomics from each patient could be utilized to give you a very valuable set of information looking at the tumor location or looking at histopathological features. By using different type of machine learning as along with the pathway analysis, you can narrow down that which of the most crucial pathways like integrin link kinase and PI3K turned out to be very crucial pathways. And then that lead you towards the potential possibilities of looking at the drug repurposing or the new drugs which can be utilized for the towards the controlling the aggressive nature of these tumor type. So I hope you are getting the bigger picture of where the small puzzles what we are giving you is going to fit into towards your your overall projects. Other thing which again I'll take a couple of minutes to just talk about another brain tumor type, which is the pediatric brain tumor. It is a medulloblastoma. It happens mainly in the children, and is still being one of the very aggressive and although it's a rare type of it's not so commonly used, but very aggressive tumor type. So what was known from the previous studies in the medulloblastoma that there are 4 different subgroups available, which includes like group 4, group 3, SHH type and Wnt type. And one of our collaborators from ACTRAC, Dr. Neelam Shishwath and her group, they did the microRNA analysis almost a decade ago. And they could show that there are 4 different patterns are being formed in these patients, Wnt, SHH, group 3, group 4. While Wnt and SHH has very distinct signatures, but group 3 and group 4 has quite overlapping signatures of the genes, quite similar in the overall expression profile. But when we look at the patient survival and the aggressiveness, then group 3 turns out to be most aggressive type and patients survive less as compared to group 4 tumor type and Wnt and SHH type. So if you look at here, there is gene signature while at the microRNA level, there is not much change, but the aggressiveness of the group 3 tumor is much more. That's where the idea was from these observation and this collaborative meetings with these clinicians, that whether we can use proteomics to find out what makes group 3 different than group 4, because so far there's no understanding that there are 4 subtypes, group 3 and group 4 are different. Group 3 is very aggressive tumor, but what makes them aggressive, have no idea. So can we look at the proteins and find out some of this information? So first thing is we followed the same standard workflow of the getting the protein from each of the brain sample, extracting the protein, digesting them, running in mass spec, generating each patient specific libraries and data set from that. It was quite huge data and pretty good quality data of 5,000 plus proteins. We could see 4 different subtypes coming in the proteomics data as well. And then each one of those analysis revealed set of protein which we wanted to take forward. I'm moving mainly towards the group 3 and group 4 comparison. That was the major interest in this particular case. And this is where the proteomics proved to be very powerful as compared to other genomic technologies, because now you can see the express changes what is happening in the dynamic proteins. And quite a few set of proteins were actually changed in group 3 and group 4 comparison. We also did validation of our study with an independent data set which we obtained from the Broad Institute that time, who was also doing this project on the pediatric medulloblastoma. And it was with very high efficiency. We could actually classify their blinded sample based on our proteins, which we thought as a good classifier of group 3 and group 4, except one misclassifier, everything was classified very well. So this was very good confidence boosting for us. And looking at this now, then we thought about what makes group 3 tumor different than the group 4 tumor. That was the clinical and biological question to be addressed to start with, right? So we found that hypoxia was one of the very key factors which is governing along with glutathione metabolism and arginine biosynthesis pathways. So some of these things we are now taking forward, investigating that what hypoxia signaling is contributing in the group 3 tumor, then looking at more of the gene enrichment analysis. You can see periostin, some of the HIF alpha factors, some of the proteins linked to the LDH and other ARAP protein, PC kinase. Some of these proteins are really mapping as a very crucial hub towards the hypoxia signaling pathways. And finally, we came up with the more mechanistic insight at how different type of changes which are happening in the group 3 tumor, how that makes them different than group 4 tumors, and finding glutamate receptors, different kinases, how they are actually driving this particular aggressiveness of the tumor type. So again, I'm not going to make it more complex for you if you're not from the bio background. For you to appreciate these parts, these are not going to be part of any questions, but rather giving you an idea how a workflow of extracting the protein, doing the entire data analysis, looking at pattern analysis, looking at pathway analysis and network analysis, then drives you towards the biology and makes you and moves you towards thinking about what could be the crucial factors which could be utilized. And of course, biology is very complex. We are still at this stage. Can we now inhibit these glutamate receptors? Can we take those cell lines and try to look for different drugs which could inhibit these and control this type of aggressiveness of the group 3 tumor type? So again, proteomics supported the subtype classification of transcriptomic study, but also we could see that there are some quite distinct changes happening, especially with hypoxia-mediated pathways in the group 3 tumors. And we are now trying to understand some of the specific type of SHH and the group 3 tumors and neuronal cell behavior, how it is important for the clinical studies. So just to summarize, I think I have been trying to give you a very balanced perspective so far about technology and biology. I'm trying to give you the technology workflow of proteomics, but also trying to give you the bigger picture idea where these small softwares and your analysis workflows are going to fit well to address the biological questions. And I think that's very important to keep an insight and some of these things, towards your PhD journey if you are interested for the research activity. And if not, at least, even if you are part of these small projects, you should know that where you are going to contribute towards while using these softwares. There are many undergrads from IT Bombay, from computer science, electrical engineering departments who have been working with us on different projects, and especially looking at the best classifiers with the machine learning approaches, looking at the best network analysis, trying to find out these pathways have been quite helpful with these students and these interns who have done the project with us. And in some of these papers, you will see if you go back, you'll find those authors which are part of our UG programs as well. So again, you can contribute a lot even if you come from different background, because you can bring in very fresh perspective of looking at data and trying to get the best of the within the humongous data set, what could be the best distinguishing data for comparing group 3 versus group 4 in this case, or looking at different subtype of the meningioma brain tumor. Of course, I have not spoken to you about a lot which we do in the lab on brain tumor, especially on the glioblastomas and all. If sometime in future we have time, we'll talk about it. But I just wanted to give you some story here to you appreciate that how Reactome string pathway analysis is crucial, linking you from your quantitative data which comes from the LFQ or TMT based workflows. So today we are going to learn more on the software analysis and the workflows from the Reactome, moving with the Gse analysis, and then which will be used for your today's quiz. Then we are going to move with the string function protein association network analysis, which will be used for your follow-up class in the next class. We will just give you a demo of this, and you can learn some of these things and use that as a homework for your next quiz activity. So that's pretty much for giving you an overview of quantitative proteomics and where we are currently. I'm open to take a couple of questions if you have any so far, and then we are going to move on to some of the demonstration of the softwares. Any questions? Any comments, students? Any one of you think you can use proteomics in any way now toward the kind of problem which you are working on or the projects which you have been assigned to work on? I'm sure all of you are at very early stage of your career, but still, if you are joining PhD journey, PhD program, do you foresee any use of these technologies or these approaches in your research? Anyone? Please use the opportunity to talk and ask questions. What happened? Is there anybody there or we are all muted? Aditya?
Yes, sir. Good morning.
Good morning. So what problem you are working on and how you foresee use of proteomics?
Sir, I am actually an undergraduate student, sir.
You are undergraduate student, but I'm sure you can still learn and contribute a lot. So which program you are in?
Sir, I am actually in material science department. I've taken it up as a minor course.
Right. Are you able to follow up some of these software analysis and the workflow which you are
Yes,
doing?
sir. Yes, sir.
Okay. Are you still finding it is okay from the engineering side or it is becoming biology heavy?
Sir, I'm actually finding it very interesting, especially, sir, since last semester I did the course on metabolomics and all. Sir, so this
Okay.
is quite related to that, and that's why I'm able to find it very interesting.
All right. Very good. Ashi, how about you? Ashi Das Gupta.
Good morning, sir.
Good morning.
Yes, sir. So even I have not worked with proteomics so far, but I am finding this really interesting, especially the softwares that we use regularly. I think it's a great learning experience that we are, even at home, we are getting to learn these softwares and we can implement them in our like studies if we ever work in proteomics.
PhD program or
Yes, sir. In chemistry department, yes, sir.
with whom you will be working?
Professor Shobna Kapoor.
All right, so yeah, so Shobna, we work very closely on many things. So I'm sure you can use some of these things in the problem of in her lab on mycobacterium or some other type of
Yes,
clinical
yes,
problem.
yes,
Okay,
sir.
all right, good. Anjali, how about you?
Good morning, sir.
Hi.
Sir, I am right now doing my MTech in biomedical engineering, and I took this course as my subject. I found it interesting. I talked to my student companions. They also, guided me to take this course because this was kind of interesting course because it has a lot of hands-on experience on this course. Other courses do not have much of that since we are sitting at home. So I thought that it would be interesting to work, but sir, sometimes it's getting a bit difficult, like sometimes getting a bit fast.
Please
So
stop us, ask questions so that we are
yes,
also.
sir. Actually, it's like so bounded. 1 hour, it's like it. I feel just it's not enough. But from last time, since we can do it at home, so it kind of I'm kind of relieved in that sense. But other than that, it was like a bit going a bit fast
No, we
earlier.
understood
Yeah.
because some of you also have network and connectivity issues. So
Yes, sir,
doing
exactly. So last
doing the
time what
two at the same
happened,
time is
yeah,
challenging.
yes, exactly, sir. So last time what happened, my screen was not changing, so I was not able to follow the Perseus one properly. So I did bad at quiz, so that happened.
All right.
But yeah,
So
otherwise it's going good.
good, good. I hope all of you are also able to follow that, since we have already showed you the instruments and the data and the workflows and the settings of the instrument, you understand by now that how data being acquired. And now when those Excel sheets comes in front of you, you should be in a position to at least visualize quickly that what all has already gone to give you that data set, right? So we have not told you about how we process the sample, that I will talk in some time, but at least running the peptides in the mass spec and generating those data, workflows, labeling, no labeling, those kind of stuff should right away now start coming to your mind, all of yours, to think about the workflows. And of course, then as we are training you on each software one by one, eventually we are going to give you some projects or the problems of problem statement and some data, and then you are going to work independently like a researcher, and you are going to give us some idea that how you can give some insight from that data set. All right, going to be interesting. Okay, so to save the time, if there is no burning question right now, we can move on to the Gse analysis, and I request Deep to take over.
Yeah, good morning, sir. So hope my screen is visible and audible.
Yes, go ahead, please.
Okay. So before going directly to the pathway analysis, I feel that you guys should understand that why we are doing what hands-on we are doing today, and how the different softwares that we are doing one after another, how it is linking and giving helping us to understand the data well. So, for example, as you have understood from the first class that we are using Orbitrap mass spectrometry or any kind of mass spectrometry to generate data set, and those data set comes in the form of raw file. Then we are using MaxQuant to understand what are the and we also using a database, for example, the human database from the UniProt, and we are trying to understand which are the proteins that got identified, and using LFQ quantification algorithm, we are also understanding the what is the intensities of these proteins in different samples, right? Then you understood and do the hands-on in MetaBioAnalyst and Perseus, where you have tried to understand and decipher what are the different, statistical analysis on the basis of the different statistical analysis, the pre-processing of the data. So in this two software, what you have done in a very brief way, first you have taken the data, understand the quality of the data. Then you have taken the data forward for different kinds of filtering steps. Then you have taken it forward for doing the normalization, the missing value imputation, log transformation, which are all comes under pre-processing of the data. And then finally, you have done the statistical test. And after doing the statistical stage, then we have learned that how to visualize the data using heat map, different correlation plots, and violin plots, right? But after doing all these also, we couldn't really understood that how do my data relate to the known biological functions, right? So for this thing, what after this analysis, what we got is that some data, for example, the uniprot ID accession, control, treatments, a lot of values we are getting, which are, transformed values and or the, normalized value, which we know as a data, the quality is good. But still, we are not able to understand that how these big list of proteins are actually having some biological understanding on how it is helping us to understand that how the disease biology is actually working. In the same way, we are getting the list of statistically significant protein, but still we are not much aware that how the things are going. So in the last class, Shashwati has given you some idea about the reactome, where all the differentially expressed protein. For example, after doing the volcano plot, the whatever the proteins that are coming from the volcano plots are all differentially significant protein, right? Because these are fold change criteria passed and p-value criteria passed. So you have taken those lists and you have tried to see how the reactome is giving the pathway based on that, right? But today I will give you a very brief understanding about the last analysis that we have done. And also at the same time, I will also give you an understanding how the GAC actually works, right? So if you come to the reactome.org, right, and if you click into the analysis tool, so the analysis tool, last time we did this one, right? Analyze gene list. So what we do, we generally take the, so for example, there are so many examples are given. Take any of the example, for example, uniprot accession list. So once this is the example data set has come, then we will click continue. We will go to the analyze, and that's it. This kind of analysis is what it does is that it tries to give you the list of pathways that mapped with the list of protein. For example, 54 proteins of that list, whatever you put as an input file, has been mapped with the disease signal transduction by growth factor, right? If you click here 54, it will show what are the 54 proteins are there. Then there are multiple different interactive, pathways are there, where if you click over here, you will get more information regarding this pathway. It will zoom into and try to show you how the pathways are linked with each other, right? And over there, as Shashwati has mentioned, that these particular yellow things that gives you an understanding that how much significance the pathways. For example, this particular yellow thing, yellow bar, is correlating with the 0 to 0.05, that is the FDR over here, or the p-value over here. That is giving an idea that how this particular thing is significantly, statistically significant. And as she told that if you go to the download thing, you can download the report and download the CSV file. So this information will help you in today's quiz to give some of the answer. And as you can see, this is a very easy way. So when you will use this particular function, for example, this particular tool, where you are putting a list of gene or list of proteins, where you already have a list of protein with you, and you are just interested to see how this list of protein, differentially expressed protein list, are having the biology, want to make a sense of the biology, right, of the different biological pathway. But GSEA, gene set enrichment analysis, is something where you are trying to upload the whole data that you have. That means you are trying to see. So let me open a demo list. So see how the data is actually looking. So this data is containing 3 control, 3 tumors. So that means the normalized missing value imputed log transform data, whatever you are getting, that means the pre-processed, the processed data, whatever you are getting of the whole number of proteins, we will use this whole list of proteins in the GSA. That means you are trying to find the alteration in the biological pathway as taking the whole proteome data into account. That means you are trying to see the major alterations that is happening and how your proteome data is helping you to understand the complete alteration of the biological pathway. So this is very strong analysis, and it gives you really good understanding that how as a whole data set or whole biology there is a alteration happening. So for example, the data set should look something like this. That means control 1, control 2, control 3, brain tumor 1, brain tumor 2, brain tumor 3. So these are the 3 groups. So if you see the chat box, you will find that Avinash has shared one of the data set. And this particular data set for this particular thing for the file GSA. So this is the same data set that you will be using for the for your quiz today. That is the GSA Reactome GSA input file. So we have shared the data set beforehand so that you are comfortable with the data set. So if you see this particular data set, you will find there is a number of healthy samples and number of tumor samples in there. So this is the same data set that you have used in the metabo analysis to do the different statistical analysis. So we are trying to keep the files same so that you can understand that if you are given a project where you have been given a file to understand and reach to the biological analysis, how to do that, right? So now let's come back to the Reactome, and now today we will do this one, analyze gene expression, right? So once you are clicking over here, so there are 3 different methods are there. The first one is PADOG. So this is something where the weighted gene analysis method has been done. That means it tries to decrease, it tries to minimizes those particular genes which maps to multiple pathways. So you might know and you will understand that one particular gene is not only map to one pathway, it can map to multiple pathway, and the common genes which map to multiple pathway might have some biasness in the biological pathway analysis. So it will tries to minimizes its effect in those things, right? And the second thing is the camera. So camera is nothing but a classical GSA approach which we will do today. And the third is that SSGSA. This is something where the expression value of a single sample based GSA happens. So these two are very much specialized one which we generally use when we know about the data set and we know what kind of analysis we want. But generally we go for camera. So camera is a classical GSA that we will use, and also make sure that you are noting down the point what we are doing so that it will be easier for you to do that. Okay. So now in the quiz, so now we will add the data set. So there are a number of different data sets are there. But I think when you will see this list, you will understand what dataset you have to choose, because the proteomics intensity, which is intensity-based quantitative proteomics data, for example, iTrack, TMT, or intensity-based level-free quantification values must be log transform. So this is the data. This is the tab which is accurately matching with our dataset, because our is LFQ dataset and our dataset has been log transform. So now we will click over here. And you have to go to that directory, wherever your file is there, and I will use the demo input GSA file. You can use the file that Avinash has shared, so that you will already have the file analyzed before the quiz, right? So now the next part is that you can see here it is giving the 3 control 3 tumors. So now we will put the annotation over here, and this part is very important. So now we will put an annotation of maybe healthy for control. Okay, and we will copy this one and paste over here, right? And maybe tumor for the brain tumor one. So what we are trying to do, we are assigning a group in the samples. So same thing you will also do for your part, right? So once this is done, then we will click continue, and over here we are designing the statistics. That means what kind of comparison we want. So as hello.
Sir, can you show that again? What you did?
Okay, so over here you were once you are putting the data set, all the name of the headers will be shown over here, right? The sample name, right? Is it showing there?
Yes, sir.
Yeah. So now what you will do, you will click the plus over here, plus option over here. So that means you are putting an annotation grouping. Okay. Now you will give a annotation groups. For example, for control, I am putting healthy because these are healthy samples. And for brain tumor, I am putting tumor. Okay, so it's not like that you have to use the same groups, but for a general terminology, I am using this. Okay, so for your case, the number of samples will be high, and on the basis of that, you will do the grouping. Okay, so for example, if I go to your thing, there are total 8 healthy are there, and there are total, I think, 8 tumors are there, right? So for example, the same thing you will do 8 healthy and 8 tumor. Am I clear?
Yes, sir.
Okay, fine. So now we will click continue over here, and we will do the step 2.2 where we are doing the statistical design. So we will select the comparison as annotation 2 because we know the last annotation was annotation 2, and we will, the first group we will select tumor, the second group we will select healthy. That means we are doing the analysis for tumor versus healthy. Be very careful in this part, because if you are doing healthy versus tumor, the pathway will be based on the healthy only, and if you are doing tumor versus healthy, so the pathway based on the tumor only. That means the upregulated pathway in tumor versus healthy will become down regulated when you will do healthy versus tumor, and based on this all the quiz thing will be done. So all these steps has been given in the quiz question, so be careful while doing checking, like putting these all these steps together, right? So now after this, we will click continue. Once this is done, so we have put tumor is the first group, healthy as a second group. We will click continue. So once this is continue, once we have clicked in the continue, we will see that this particular data set has already been uploaded. If you want to do some editing, you can click over here and you can go back to the edit annotation. Right, and after that we will click continue over. So it is asking, for example, if we go for big data set, multiple data set, and we know that Reactome takes some time, so you can put your mail ID, so that once the analysis is over, it will ping you back in your mail that the analysis is done. But for, as we know, this is a small analysis, so we don't need to, we will not put any mail ID over here, and we'll just simply click go. So once you are clicking it go, you can see the data submission, and followed by the analysis will be done, and it will take maybe a couple of seconds, minutes time, and it will be done. So you can see the performing analysis is 90%. We are creating.
Sir?
Yeah. Yes, yes.
Sir, what did you say about the annotation part? Like in my thing, it was showing annotation 1. So I selected,
Right. Yeah, fine.
in first group, I selected healthy, and in second, I selected tumor.
Yeah, so if you are selecting first group healthy, then the healthy versus tumor pathways will come. Okay. And but if you are selecting tumor first and second healthy, so tumor versus healthy will come. So you can understand in the first comparison, the pathways which will be upregulated in healthy. In the next comparison, it will show downregulated. Right. So be very careful in the quiz what first group and what second group has been given. Otherwise, your question will be wrong. Okay. Okay, so once this is done, so make sure that you are downloading few things over here. For example, PDF report you can click. Once you are clicking the PDF report, it is giving you, redirecting you to the PDF report over here. And you can also download the XLS file. So for example, sometime if you clicking over here, it will not get download. So it's better that you click a right click, save link as, and save it in your directory by giving some Gse output, something like that, so that it you can go back and check the thing. So mostly we have tried to give the questions based on the report only, but still download the GSA output. It might help you for giving some of the answers easily, right? So once this is done, we will click cross over here. Okay, so once we are clicking cross, we are getting the result output in the portal. So over here, first you will see that the whatever data we are getting for the normal Reactome analysis, we are getting almost same, but there is something over here which were not coming in the normal Reactome, right? So what it is showing is that on the basis of the data, this particular pathway is whether down-regulated or up-regulated, right? For example, in volcano or fold change, you used to see that one particular protein or one particular gene is up-regulated or down-regulated. Now using GACA, because it's a gene set analysis, that means the whole gene of that particular pathway has been used to understand the overexpression or the like up-regulation or the down-regulation of the whole pathway. So what does this 2* mean? So you need to drag this thing a little down, and you will found over here that upregulation and downregulation. So if you click, put your cursor over here, it will say that these are significantly upregulated. So the next one is not significantly upregulated. That means this is upregulated, but it is not passes the statistical criteria. That's why it is not significant. So these are not found. then these are non-significantly downregulated, and these are significantly downregulated. So be very careful about the annotation, because on the basis of this only you need to understand which pathways are upregulated, significantly upregulated, downregulated, or significantly downregulated, right? So over here you can see these informations has been given very thoroughly, and if some of you have not downloaded the PDF during that time, so again you can come back to download and you can download over here. So you can also see there are 121 protein or features which has not found. That means these particular proteins are those proteins which has somehow not mapped in the biological pathway databases that the Reactome is using, or some of the proteins has not been annotated even, right? So there are 121 proteins are there, but when we have given a list of almost 1200 protein, 121 protein will not make much differences, right? So now we will go to the Reactome pathway PDF. So what it is showing? So first, this particular volcano plot is giving you the list of those particular pathway, like the number of pathway that has passed the cutoff. And it is telling that there are total 1614 pathways are there, and 1247 fold changes of for proteins or genes are there. That means this mean number of proteins and genes has been passed the fold change criteria cutoff. So if you are coming down, you will see that there is a top regulated pathways are there. That means the citric acid TCA cycle is the maximum top regulation pathway, which is showing a upregulation, and the FDR is less than 0.01. This is the average fold change, and this is the number of genes that is associated with this pathway has been found in our data. That means 76 proteins from our data has been mapped with the TCA cycle. So like this, you will get a lot of information over here, but it will give you information of only few top 15, top 20 pathway, not more than that, right? Because if you want to get the whole list, then we need to go back to the Gse output, which I will show. So now if you come down, you will see now the protein and the gene level result. That means it will show the volcano plot, and what are the total number of proteins are there. If we are taking a adjusted p-value criteria, what are the proteins are there which has came. So there are two terms you will know. One is p-value and there is adjusted p-value, which is like almost same as the q-value. So p-value means, as you know, that there is a chance of, we are giving a chance of 5% error. However, the adjusted p-value is that in your significant list which has passed p-value, we are giving a chance of 5% error in the significant list. So as you can understand, then adjusted p-value is much more significant than the p-value, right? So this is the whole output result, as you can see in the PDF. And if you see the click into the Gse output CSV result, then you will find that there is a list of all the pathways are there, whether it is a down regulated or up regulated, this is there. But make sure we are not able to understand whether it's a significant down or significant up, right? It is only telling that up and down. So for that, we need to go back and check the portal, that how many up and up, how many down are there. Then there is other informations are there. For example, the FDR proteomics intensity. So these are the information based on the p-value, FDR, and all these information. So number of genes, how many number of genes are there, which has been mapped with the pathway, average fold change of the proteomics intensity. So number of informations are there, and make sure all these information, whatever you are showing, are based on the groups that you have assigned. For example, it is all a tumor versus healthy, because we are assigned the first group as a tumor and the second group as a healthy group, right? I think the all this information you have followed properly, you will be able to understand the Reactome GSEA, and you can give give all the question from the GSEA quiz correctly. But make sure that apart from quiz, this particular like analysis tool is very strong and is much more reliable when we are talking about normal Reactome analysis, because you are giving the the tool the whole set of protein whatever you got. So it will do the analysis by its own, and it will give you an idea what are the pathway that is coming up regulated, what is the pathway that is coming down regulated, right? So is there any question regarding this part? I think it is a pretty clear and pretty easy software which you can see. It's just you need to understand the concept that why we are using GSEA, right? So now I think you can now tell that from here, from the biological, from the significant pathway list or the whole list of processed file, what are the biological pathways that are coming. So now I don't think these question marks you will have, right? Okay, so I
All
think
right.
if it is
See,
done, yeah.
so we can
Okay.
actually give a quick overview of the string now, and then we can move for the quiz. So today's quiz will be only based on the Reactome only.
Yeah, Reactome and Gse. Yeah.
Right, but we are also going to show briefly the string so that that can be used in the next class. All right, let's move on to string. Abhilash, you want to take over?
Yes, sir. Thank you. Okay. Fine. So as we showed, you put your significant protein list and all the files actually for Reactome data analysis, and you found some protein list or some pathways from your analysis, and you can be happy with that to know the list of proteins. But now, how to visualize those list of proteins and how to interpret those results? So to do that, we use string. Okay, so we have already added the demo file for string in the analysis. Now you don't have to perform the analysis with me, but for the timing, I will just show how to perform this analysis, how to do the string. Okay, so this is the protein list. This is the significant list of proteins we are starting. So unlike the GSA, where we put entire data set with a distinct group, now we will just take the significant proteins that we have performed from the statistical analysis. Just select all the proteins, copy them, and go to string. Okay. And here, since we are choosing multiple proteins, we can select the portal of multiple proteins, just paste it, and we'll have to select the organism. Here we have this is the data from the healthy in human versus the brain tumor. So we can choose the organism human. Taking some time. Okay, now search it. So in this way, if you have UniProt ID also, it will give you a protein name or gene name. So if you want to convert the ID into gene name, also you can do just by downloading the mapping. It will give you a Excel file with all the gene name. So initially we put all the UniProt ID, and at the same time you will know the string ID, you'll know the gene name and their annotation and where it is located and all the information you'll get from here. Then we'll just click continue. To know the mapping, to know the protein-protein interaction, and to know which pathways they are belonging. So once you get the PPI or protein-protein interaction, go to string. If you want to know the full string network, it will give some hypothetical interaction. But if you want to know the evidence-based interaction, whether they are physically interacting or not, you can also choose that. So for the timing, if we choose a physical subnetwork, it will remove some of the network or edges that is not interacting directly. So if I update, you'll see only proteins that interact with one another physically or directly. But if we choose full string network, you'll get the file that has some hypothetical interaction, that is, or they are interacting through some other interactor. Fine. And here you have all the data, like the active interaction sources, whether it is from the text mining, whether it is from the experiment or databases or co-expressions. On the basis of that, you can enrich this protein-protein interaction. So if you want to get more confidence, you can choose the experiment only. So for for this, the data, the interaction will be only from the published paper. If you want to hide this network, if you don't want to show the interactor which are not mapped, which are not interacting, also you can hide them by just clicking the hide disconnected nodes in the network. So this is just for visualization, but the most important thing comes in the analysis tab in analysis portal. If you click the analysis, it will keep the different ontology databases and what are the different pathways coming from here. So mostly to get more confidence, we click KEGG pathway or Reactome pathway. So for the timing, if I click the KEGG pathway, it will give the pathways or the network that are mapping or that have the that are mapping to the KEGG database. Also, you will see how many proteins are mapping from here and what is the strength of the node, strength of the network along with the false discovery rate. So all these factors, all these things are important to have to get more confidence on this. So if I click here, it will annotate with a proper coloring so that I'll know which protein or which proteins are belong to this pathway. So if you have a hypothesis before, and so if you have done some kind of treatment drug assays and you know where your drug is working, on the basis of that you can choose which proteins from this pathways are getting mapped, and on the basis of the proteomics data set you can check their abundance and you can find whether this node is upregulated or downregulated. So I have chosen the citric acid cycle, and here I found that most of the proteins that are belonging to this pathway is colored in red. Accordingly, you can choose different metabolism, different pathways to know about the mapping of this. And later you can make a cluster and you can analyze them to have more confidence on the pathways along with their interactor. And if you have the abundance, you can easily tell that this pathway is either it is upregulated or downregulated. Okay, so we'll discuss this more. Discuss this more in next class. But yeah, once you have done this, you can also download the different files from this analysis. If you want to know the image, you can download. You can export the image. Also, if you want to do further analysis, further enrichment, further analysis using Cytoscape for the visualization, you can download the SVG file. You can export it into Cytoscape for better designing and better understanding.
All right, Abhilash, thank you for giving this overview. And again, some of these are very user-friendly softwares, which can be, one student will start playing with them, they can start feeling more comfortable about it. So we have already given some file for practicing about it. All the students, please do try out a string when you get some time, and make sure that you are comfortable looking at the data output. And more you will play with these, whether Reactome or string. Even there are other tools like David and Panther and many others are there, which actually can give you much better visualization of your data, much better sense of the data, how you can see where most of the changes are happening, which of the pathways and which of the key proteins and networks are being involved in the hypothesis which you have. So these are towards connecting your abstract data to the more dynamic biology. All right, we'll stop here for this demonstration and this session today. We are now going to have the quiz, which will be based on the file which we have already shared earlier on the Reactome and GSEA. So, Shashwati, could we start the quiz now? And time will be 15 minutes or 20 minutes, Shashwati?
Yes, sir.
How much time? 15?
20 minutes.
20 minutes. That's fine. All right, everyone, good luck. Let's start the quiz. In between also, if there's a mistake which you get stuck, do ask questions on the chat. We'll be happy to address that further. Okay. All right. That's all for the lecture. After this, you can leave. Thank you.